The overall goal of this methodology is to perform a 3D in vivo morphological assessment based on an ultrasound analysis of human muscle tissue.
This method is a promising technique for answering key questions in healthcare and sports about diagnosis and follow-up of muscle function after treatment and training.
The advantage of this technique is that measuring the muscles by 3D ultrasound is a fast and cost-effective alternative to magnetic resonance imaging.
This 3D ultrasound technique provides a non-invasive method for determination of muscle volume, fascicle length and the physiological cross-section area of skeletal muscles in vivo.
Since 1980 we've been active studying the form-function relation of muscles, first in human cadaver muscles and making mathematical models of those muscles, and then in animal experiments. With the improved ultrasound technique, we will now be able to study human muscles in vivo.
To assess the gastrocnemius medialis muscle, or GM, first, have the subject lie prone on an examination table with both feet over the edge of the table. Place a support under the tibia to align the lower leg horizontally, and fix the thigh to the examination table with padded lashing strips to prevent knee extension during the experiment. Fit the foot of the leg to be scanned into a custom-made foot plate, and connect a custom-made torque wrench with a goniometer attached to the foot plate. Find the foot plate angle.